Hi, this is Mrs. Nagayama, and this recording will help you with quiz 1.05. Please review sections 1.01, 1.02, 1.03, 1.04, and 1.05 before taking this quiz. So this quiz is about the scientific method. Scientists go through an organized set of steps to answer questions. Taken together, those steps is what we call the scientific method. We'll be going over each step and going over sample questions from the quiz. So step number 1, making observations. So the scenario we're going to be discussing today is imagine that we have a scientist, and that scientist is growing colonies of bacteria in petri dishes in her lab. A petri dish is just a clear dish with some medium in it that, like you see in the picture. Each little yellowish or red dot represent a colony of bacteria on the petri dish. So what she notices is that the petri dishes closer to the window have more colonies than the ones that are further away from the window. So that's the observation that she made. So the step number 2 would be to ask questions. So after you make observations, you can now ask questions based on your observations. So the scientist can ask, what are the factors that promote increased growth of bacteria? Is it sunlight or is it temperature? So you're thinking about why is it that the ones that are closer to the window have more bacteria than the ones that are further from the window? Is it because there's more sunlight near the window, or is it because it's hotter near the window? So that leads us to step number 3, forming up a hypothesis. So a hypothesis is a possible explanation for an observation based on information and knowledge that you have. The information and knowledge that you have, you will get from your observations. So to kind of sum it up, in order to form a hypothesis, you need first observations, you need to have some sort of criteria that you're going to follow, and then some sort of reason or kind of explanation of why you think something is happening. And then you can put all of those components together and form an if-then statement. So if a certain criteria is present, then this must be why it must be happening. So an example of a hypothesis for this scenario would be at the bottom. If there are more bacteria colonies in the petri dishes that are in a warmer environment, then bacteria colonies grow fastest at warmer temperatures than bacteria colonies do in colder environments. So in order to figure out what a hypothesis is, you need to make sure that it's an if-then statement and that it actually answers your question that you have. Step number 4. Then what happens is once you form your hypothesis, you can then perform experiments to test your hypothesis. So for her experiment, the scientist put 40 petri dishes in the dark, keeping 20 dishes at 80 degrees and 20 at 65 degrees. All other variables are kept constant. So why do you think it was important for Sally to keep the 40 petri dishes in the dark? We're going to go ahead and explain that on the next slide. So when you are performing an experiment, you only want to test one variable. So we have the independent variable and we also have the dependent variable. The variable that you are testing is going to be called the independent variable. So in this case, it would be the temperature that the bacteria are placed in. Are they placed in a warm temperature or are they placed in a cooler temperature? The dependent variable will be the variable that is measured. So we're changing the temperature that the bacteria are in, and what we're actually measuring, which is the dependent variable, is the number of bacteria colonies present. So step number 5, in order to really know whether your experiment is going successfully or not, you need to collect accurate data, and then after you collect all of your data, you will then analyze your data. So if you think about what kind of data should Sally collect for her experiment? So her experiment, she is testing whether there are more bacteria colonies near in warmer temperatures or than cooler temperatures. So what kind of data would she collect? In this experiment, the data would be the number of bacteria colonies. The average number of colonies in each dish at the higher temperature was 30, and the average number of colonies in each dish at the lower temperature was 17. So now Sally can look at that data that she has and analyze it so she can come up with a conclusion. So first she collects the data, then she'll analyze it. So when we collect different types of data, first we can collect qualitative, and second we can collect quantitative. So qualitative data are data that is descriptive, usually using adjectives. So if you look on the left-hand side, I have a picture of pencils, and I want you to think of different ways that we can collect qualitative data about these pencils. So what are some adjectives that describe these pencils? So some that can be mentioned are that it's red and that they have sharp points. Some quantitative data would be data that is measurable, so you would be using numbers. So we can either measure the pencils, which would be 6 inches, or we can count the number of pencils, which would be 4. So the reason why scientists usually use quantitative data is because you can't refute it. It's always correct. No one can tell you that it's not 6 inches because it clearly is 6 inches based on the ruler and the pencils. However, if you were to tell someone that the pencils were sharp, they could tell you, well, I wouldn't consider that sharp. If we sharpened it a little bit more, then I would consider it sharp. So qualitative data tends to be a little bit more relative. All right, step number 6, scientists form conclusions about hypotheses based on data analysis. So after we collect the data and then analyze it, we will then form a conclusion. So in this case, after analyzing the data, the scientist concludes that bacteria kept at 80 degrees, which is the hotter temperature, grow faster than bacteria kept at 65 degrees. After you make your conclusion, then what scientists do is they find answers to questions, and new observations lead to new questions. So basically what happens is they can now form new hypotheses based on the results of their experiment. So here's an example. The scientists first noticed the effect near a window. Another line of questions about what affects bacteria growth might involve experiments with varying amount of light. The scientists may also wish to determine the upper and lower ranges of temperature that will allow bacterial growth, or find out precisely what temperature will allow maximum bacterial growth. She will form new hypotheses and test them. So now, instead of just thinking about the temperature, she's also going to think about the light and make a test for that. Step 8 is that scientists will then communicate the results of the experiment to others. Why do you think it's important to communicate your results? Why is it that the scientist shouldn't just keep it to themselves? Well, when the scientist shares her findings with other scientists in the lab, then other scientists could design future experiments with when scientists design future experiments with bacteria in a lab setting, they could think about it like maybe we should make sure that the temperature is constant, because obviously bacterial growth depends on the temperature. So it kind of gives them more information about how they should design their experiments in their lab. And then step 9, we have once scientists formulate theories only after the hypothesis has been tested multiple times. So scientific theory is not a guess, it's an explanation for an extensive body of facts. A scientific theory is made only after many people have repeatedly tested a hypothesis in many areas of the observations in question. Okay, so here are some sample questions. Which step in a scientific investigation would a scientist usually do before she performs an experiment? A. Analyze data. B. Form a hypothesis. C. Conduct research. Or D. Communicate with others. Remember, a hypothesis is a possible explanation for an observation based on information and knowledge, so it relies a lot on your observations. So which step in a scientific investigation would a scientist usually do before she performs an experiment? It would be to form a hypothesis. Remember, you always make those observations, form a hypothesis, then you can perform the experiment. Here's another question. Which statement best describes qualitative data? A. It cannot be represented by numbers. B. It cannot be a dependent variable. C. It cannot be controlled. Or D. It cannot be analyzed. So remember, qualitative data is the one that we use to describe the quality, and usually we use adjectives. So here's the slide that we had for qualitative versus quantitative. For qualitative, we use descriptive words like adjectives, and quantitative, we use numbers. So the best answer for this one is that qualitative data cannot be represented by numbers, which would be choice A. All right, here's the next question. A scientist spends 3 months collecting data during an experiment. Which of the following steps is important for him to do next? A, form a conclusion, B, form a new hypothesis, C, communicate the information, or D, analyze the data. So far what he's done is collect the data. Now what should he do? Remember, step 5 was that scientists collect accurate data and then they analyze it. So the best answer for this one would be D, analyze the data. So that concludes all the sample questions from quiz 1.05. If you have any questions, feel free to email me or contact me, and I can give you more resources to help you be successful on this quiz.